Welcome to this video on mitochondrial disorders. I'm your course instructor Tendral. Today we will dive into how defects in the powerhouse of the cell, the mitochondria, can lead to a range of diseases. We will explore a case study, look at the key symptoms, diagnosis, and potential treatments. Let's begin with the introduction to mitochondria. Mitochondria are essential for energy production in cells. They generate ATP through oxidative phosphorylation. Interestingly, they also contain their own DNA and can reproduce independently, while mutations in the mitochondrial DNA or nuclear DNA affecting the mitochondria can lead to serious cellular energy deficits. So we will see the mitochondrial disorders related to this. In the illustration, you can see the structure of mitochondria, showing the outer membrane and inner membrane of mitochondria. You can see the intermembrane space between the inner and outer membranes, and then the DNA in the matrix. So this is the structure of mitochondria. Now let's see the overview on the process of ATP. You can see the glucose entering the cell membrane from the extracellular surface into the intracellular surface of the cell, where glucose is converted into pyruvate through the process called glycolysis. And then this pyruvate enters the mitochondria, where the pyruvate is converted into uh, carbon dioxide, and the NAD plus molecule is converted to NADH, which is a reduced electron carrier, donates electron to the electron transport chain, which is present in the inner membrane, that is inner mitochondrial membrane, initiating oxidative phosphorylation, which will lead to ATP synthesis. Now let's see what are mitochondrial disorders. Mitochondrial disorders are a diverse group of genetic conditions caused by problems with energy production. These disorders can affect many systems in the body simultaneously, especially high energy demanding organs like the brain and muscles. The mitochondrial disorders can be genetic or acquired. Now let's see a case study. Let's examine the case of a 9-year-old presenting with muscle weakness, fatigue, and developmental delays. Her mother has reported similar, though milder symptoms. This familial pattern raises suspicion of a maternally inherited mitochondrial condition. Let's see the diagnosis and testing for the case study. Initial blood test showed elevated lactate levels, suggesting poor mitochondrial function. A muscle biopsy revealed ragged red fibers, which is a characteristic of mitochondrial disease. Genetic testing confirmed a mutation in the MTTL1 gene, commonly associated with MELAS, which is mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. Let's let's see MELAS in detail. Our primary case study is MELAS syndrome, a well-known genetic mitochondrial disorder. It typically begins in childhood and manifests with neurological and muscular symptoms due to poor energy metabolism. The symptoms include muscle weakness, seizures, vomiting, stroke-like events. The main cause is the inheritance, that is maternal DNA, that is maternal mitochondrial DNA mutation. Here is an illustration showing the normal and MELAS conditions. Here you can see in the normal condition, the translation includes the codon UUG, while the MELAS condition showed a specific defect in translating UUG-rich gene. Beyond MELAS, there are numerous mitochondrial disorders, each with unique symptoms. Leigh syndrome affects the central nervous system. Murph presents with seizures and muscle issues, while Kearns-Sayre syndrome deals with eye muscle weakness and heart conduction defects. And Leber's hereditary optic neuropathy shows sudden vision loss in young adults. Not all mitochondrial dysfunction is inherited. Some are acquired disorders, results from the exposure to toxins, aging, or any chronic diseases. These disrupt mitochondrial performance, leading to similar symptoms as genetic disorders, but with different origin. In the illustration, you can see the UVR, which increases the oxidative stress, leading to ROS production, that is increased ROS production. ROS stands for reactive oxygen species, which are highly reactive molecules containing oxygen formed by byproducts of cell metabolism. And this plays roles in both beneficial and harmful processes, but increase in ROS production leads to harmful conditions. And then you can see the organelle dysfunction, damaged mitochondria, and then altered gene activity due to the aging, decreased antioxidant, and accumulation of mutation. All these lead to oxidative stress, which is damage due to free radicals. Diagnosing mitochondrial disorders can be complex. It often involves a combination of genetic testing, biochemical assays, and imaging techniques to evaluate energy production and tissue structure. While no cure exists for the mitochondrial disorders, several treatments aim to manage symptoms and slow disease progression. Supplements may boost mitochondrial performance and Gene therapies are under investigation for long-term solutions, but currently there is no cure for mitochondrial disorders. In summary, mitochondrial disorders form a complex and evolving field of study. While many of the mitochondrial disorders are inherited, others can be acquired throughout life. Understanding both types of mitochondrial disorders is key to developing effective diagnosis and therapies. Continued research into mitochondrial biology offers hope for more targeted and permanent treatments in the near future. Thank you for watching. See you in the next video.